high school in Putnam City. I guess it's part of Oklahoma City. So Lucas went to high school in Putnam City. He is a junior at Oklahoma Baptist University, where he's studying chemistry and math. He spent the summer working in the lab of Courtney Montgomery in the arthritis and clinical immunology.
Thank you, Dr. Kinker. Today I would like to speak with you about how genetics, specifically two genes called ID3 and IFIH1, impact kidney health and autoantibody production in lupus. I'll start by giving you a bit of background, which will hopefully include everything you need to know in order to understand my project. Then I'll give you my questions and methods of research and end with the results and probable implications of those results. So what is lupus? Lupus is an autoimmune disorder, meaning your immune system produces antibodies against your own organs. It's a highly variable disease. The American College of Rheumatology outlines 11 criteria that are used to diagnose lupus, pictured here. But a person only needs to meet 4 of those in order to meet a diagnosis. So 2 people suffering from lupus could look so different that you wouldn't even guess they have the same disease. But what they do have in common are pathogenic or disease-causing autoantibodies. And you know when a microbe enters your body, like the flu, your body will produce an antibody to help get rid of it. Well, autoantibodies are those haywire immune players that your immune system produces, then go to town on your own organs. And they can be very destructive. They are the primary, they're the underlying cause of many of the symptoms of lupus. And that is why they're one of the focuses of my project. Also, this aberrant immune response does not have a cure. You simply treat the symptoms of lupus as they arise, whether that includes getting an organ transplant or blood transfusion or taking immunosuppressants. And because you're on immunosuppressants, one of the common causes of mortality in lupus is third-party infection. Lupus can also lead to heart disease and commonly kidney disease. In fact, kidney disease is one of the more serious and more common manifestations of lupus. And in that case, we call it lupus nephritis. And that is why it is one of the focuses of my presentation today. So who has lupus? Well, about 20 to 150 people per 100,000 have it, depending on what sort of demographic you're looking at, because it varies quite widely from ethnicity to ethnicity. Minorities are afflicted more often, and it's often more serious in minorities, including vital organs more often. It also afflicts women of childbearing age primarily, so you can imagine that it can be quite debilitating to suffer from a condition for the greater part of your life. And as far as we know, there are environmental and genetic causes of lupus. But where and how genetics are influencing lupus in this continuum from normal immunity to organ damage, and where and how the environmental factors like drugs, viruses, and UV radiation are, we do not know. We simply know that they do impact them. Today I want to focus on the genetics behind some of the symptoms of lupus and hopefully elucidate more of that genetic basis. So I'll give you a few simple terms that you'll need to understand my project. I'll talk a lot about genotype. And when I'm talking about genotype, I simply mean your genetic makeup, the thing in your DNA that makes you different from other people. And today I'll speak primarily in terms of single nucleotide polymorphisms. I may call them SNPs or markers or variants or mutations, but I mean the same thing. And it's essentially that portion in your DNA where you look different from someone else at just one base. And as you can see, people also have phenotypes. In this case, the phenotype is, purple, blue, orange. But the phenotype is essentially an observable manifestation of your genotype plus the environment. So, just because 2 people have the same genotype, they may not have the same phenotype because it's a combination of environmental and genetic factors. But the genetic factors do come into play. And today, of course, we're not going to be looking at colors. We're going to be looking at things like the amount of protein in a person's urine or the amount of autoantibody in their blood. Now, there are 2 genes we're going to be looking at today, And we're looking at them because they've been of particular interest in the past. The first is called ID3. It is known, it's a transcription regulator. And when something goes wrong with a regulator DNA transcription, it's hard to predict all of the ramifications of that. But we do know is that ID3 plays a role in inflammatory disease, and it's been linked to kidney health in atherosclerosis patients. But atherosclerosis is a kind of heart disease. The second gene we're going to be looking at is IFIH1. IFIH1 it plays a role in a viral recognition pathway. So it's understandable that it could be linked to these unusual immune responses. It's been linked to things like cell death, those lupus-like kidney problems, and autoantibody production. So it's a particular interest in my project. The phenotypes we're looking at, like I said, are not colors, but they are different measures, such as the renal phenotypes, which are measures of how well your kidney is operating as a biological filter. And we have the autoantibody measures. Our autoantibody measures include the simple presence or absence of these 7 autoantibodies and the actual quantitative levels of those antibodies. We want to take both into account. So what I did with my project is I used statistical methods to perform association testing. And essentially what we're asking when we perform association testing is, do people with the same genotype have similar phenotype values? So when we see something like this, where we have 3 genotypes, AA, AG, and GG, we want to see a linear relationship like we see here. And when we do see this linear relationship, we use statistical methods to get a p-value. And essentially what the p-value is it's the probability that that linear relationship we're seeing is there simply by chance. So we want the p-value to be extremely low. That way, we can be certain that that linear relationship is because of a true biological fact. So we performed some preliminary testing, and we saw 3 suggestive SNPs. Suggestive meaning that we didn't necessarily see statistically significant results, but we did see results that suggested that there may be something there if we just had a larger sample set. So we genotyped 163 people of these 3 SNPs. I joined forces with Adam, and he was an incredible teacher and easy to understand. He's a good guy. And you'll have a show coming on Fox this fall. So, on to the results, which is the exciting part. First, I'm going to cover the results over ID3, and then I'll cover the IFIH1 results. With ID3, we didn't see any significant associations with the kidney measures, but what we did see were some interesting numbers with the autoantibody production. So what you have here is called a zoom plot. And essentially you have the SNPs along ID3 plotted according to position, and you have them plotted against the negative log of their p-value for their association with the specific phenotype. So in this case, this is the negative log p of this SNP with nuclear RMP presence. And we're looking for a p-value, in this case, below 0.00125. So we want a negative log p above about 2.9. And we saw 2 SNPs in particular that showed significant results, or at least this one was a suggestive result. The first being RS11574. And RS11574 showed results with the mere presence of nuclear RMP. So we wanted to see what exactly was happening. So we looked at the measures and plotted them here, the genotype and what the percentage of people who were positive for that autoantibody. And what we found was interesting, that actually the T is the minor allele at this particular SNP, and it appears to have some sort of protective effect. You're less likely to have nuclear RMP antibodies if you have the minor allele here. But even more interestingly, the heterozygote seems to be more protected. So there seems to be some sort of emergent codominant effect here that would probably require some in vivo or in vitro experimentation to really elucidate why that's happening. Here, we saw another SNP called RS1555026, and its association with the actual amount of nuclear RMP. So you can see the plot here that a person who has the minor allele, which is in this case a C allele, appears to be more likely to have higher levels of nuclear RMP. Of course, we have some limitations on our results because we don't have any people who are homozygous for the minor allele, but the trend does seem to be there. And interestingly enough, we saw a similar trend with another autoantibody called SNP. So we were wondering if SNP and nuclear RNP autoantibodies are related. And what we found was that they actually are. And the fact that the coincidence of nuclear RNP and SNP autoantibodies is not necessarily new. It's, we're merely corroborating what has already been established in previous studies. But something that is interesting is we found that not only do they typically occur together, but their titers, their quantitative levels are closely linked in this linear sort of fashion. But what I think is really interesting here, and what I want to emphasize, is the color scheme. Because we have red dots and black dots, and the black dots are the people for whom we have a genotype at RS1555026, the SNP that we think might be associated with these titers. The people who are represented by red circles are the people for whom we do not have the genotype. And you can see that we have some very interesting people with high levels of both autoantibodies, and we don't even know what they look like at the SNP that we think could be affecting those autoantibody levels. So a future direction for this project could include genotyping those people and seeing if that association we think we see becomes even stronger the more people we genotype. But I do think that this has extreme promise because of the biological viability. Going back to the two SNPs, it's interesting to note that RS11574, which is associated with the mere presence of these two autoantibodies, nuclear RNP and SNP, is a missense SNP, meaning it actually changes the structure of the protein produced by the ID3 gene. So depending on the mutation you have here, you can have one of two different functioning ID3s. And interestingly enough, this dichotomous change in function is linked to this dichotomous measure, this absence or presence of nuclear RNP. Now, on to RS1155026. It's actually not in ID3, but it's just upstream of ID3 and actually in an enhancer region for ID3, meaning that it's likely this mutation could alter how much ID3 is expressed. So, if you have the structure of ID3 determining whether or not nuclear RNP is present, and the expression amount of ID3 determining how much nuclear RNP is produced, it's a rather compelling biological story. So I do think that this project has promise and strong future direction. Now remember, we also looked at another gene, IFIH1. With IFIH1, we performed the single variant analysis, trying to mark one SNP to each, SNPs to each phenotype, but we didn't actually see much with the kidney measures or with autoantibody measures. So we tried a different route in which we considered the interactions of IFIH1 with other genes within its pathway. And I know this looks really busy, and you can see there's a lot going on here, but what I really want you to focus on is this. We have two proteins, RIG1, which is produced by DDX58, and MDA5 produced by our gene of interest, IFIH1. And you'll notice that this is a viral recognition pathway, and in this pathway, these two proteins produced by these genes are playing a very similar role. They're both recognizing viruses. And they're actually what we call paralog genes, meaning they have very similar structures and are probably of a similar evolutionary past. So you can see that they recognize viruses, then they send a signal down the pathway, and eventually it arrives at inflammatory cytokines, which cause inflammation, as the name suggests. And if this immune response is actually improper or haywire, it can cause organ damage. So we found that there were mutations within IFIH1 and DDX58, its paralog gene, that in their interaction influence creatinine clearance levels. And creatinine clearance is a very reliable measure of kidney function. So it seems as though if you have mutations in both of these viral recognition proteins, then you are likely to have organ damage or kidney damage, and that this pathway could be linked to that sort of organ damage we see in autoimmune disorders. So, conclusions. It appears as though ID3 may influence autoantibody production, But we need a little more data. We need to genotype a few more people to be certain of this, though it does show biological promise. And we noticed that nuclear RNP and Smith autoantibodies are closely linked and highly correlated, which is an interesting find. And also mutations within the RIG1-like receptor pathway, that viral recognition pathway, specifically at IFIH1 and its paralog gene, may influence aberrant immune responses and lead to kidney damage in autoimmune disorder. I would like to thank the people of my lab who have been with me every step of the way and helped in more ways than I can list. I'd also like to thank Dr. Fred Rhodes of the Putnam City Schools Foundation for being my donor and funding my time here. I'd also like to thank OMRF for providing the program and welcoming us Fleming Scholars in with open arms, and Carlisa and Crystal for organizing the program and making sure everything went smoothly and was enjoyable. And I'd like to thank my university, Oklahoma Baptist, for supporting me through the application process and preparing me for where I am now. Thank you.
Questions? So I guess what always surprises me in y'all's statistics is the p-values. As a lab scientist, we like 0.05.
Yes.
And so that means we think there's only a 5% chance that the null hypothesis is correct. I mean, how do y'all pick out 10 to the minus fifth, 10 to the minus third? That means there's less than 0.1% chance of it being correct.
Well, it's interesting because in this case, well, specifically regarding IN3, we were looking at 40 variations within IN3. So the more variations you look at, the more likely you are to see that really good looking p-value simply by chance. So we do find those really good looking p-values, but we have to correct for that multiple testing. So ultimately the p-values may not really be the times 10 to the minus 8, but they're significant for our purposes. Yeah.
Yes.
So can you tell me where the variant is in the structure of the ITS3 gene?
Where the variant is?
Yeah.
Well, RS11574 it's actually in an exon of IN3, and in coding it affects, it causes an alanine to threonine shift in the C terminus of the
Is
protein.
that in the helix to helix region, or is it
It is,
outside?
it is in the binding domain. So it changes the binding structure of IN3 when it's binding its substrates.
Do y'all ever consider combining a couple of these factors, a multi, if they have both of them or one or?
We actually did. We performed what we call haplotype testing, where we're looking at different combinations of SNPs within the genes and seeing how they might influence phenotype together, but we didn't see anything significant with those.
Any other questions?